Hello, and welcome to the Disrupting PFAS podcast. I'm your host, Jeff Hale. For this season of the podcast, I've curated 4 episodes focusing on the detection, destruction, and sequestration of PFAS using novel materials or processes. Though emerging technologies are promising for the ex-situ treatment of PFAS, subsurface applications are needed for PFAS remediation. Dr. Kurt Pennell and his team from Brown University have been doing exciting work with the subsurface application of materials commonly used for ex-situ treatment. This includes in-situ sequestration of PFAS using ion exchange resin. This research is especially relevant to the theme of the podcast because it involves both a novel process and material. So let's learn more about the technology from one of its innovators. Welcome to the podcast, Kurt.
Thank you. Nice to be here.
So I understand you are the 250th anniversary professor of engineering at Brown University. Could you please tell us about that distinction?
Yeah, my wife thinks I'm 250 years old, but that's when the school was founded, so that's how it ended up that way. And the donors are anonymous, so that I guess they settled on that. So I came to Brown about 4 years ago. And I set up my lab. I basically have 2 types of labs. One is a mass spec lab. We do a lot of the analysis for PFAS in that lab using high res, so we can look at both targeted and non-targeted analysis. And then we have a more, what I call a more like soils or wet lab where we run our experiments, and we do the analysis in a separate lab.
Very interesting. Could you please talk about some of the other positions you've held before joining Brown?
Sure. I did a postdoc, and then I was a research professor at Michigan in the civil and environmental engineering program. And then I moved to Georgia Tech in '95, I think, right before the Olympics. So I got to experience the Olympics in Atlanta, which was nice because it was right on campus. And I went through basically assistant, associate, and full professor. And then in 2009, I left Georgia Tech to go to Tufts to be department chair of civil and environmental engineering. And I did that for about 8 years. And this opportunity then arose at Brown. I really wanted to get back doing more research rather than administration. And so I sort of shifted back, and now I'm pretty much focused on my research. And I teach undergrad class, intro to environmental engineering, and then a grad class on fate transport remediation. Mostly subsurface, in situ. Try to focus on in situ processes. And I sort of lead the environmental engineering program here at Brown.
Excellent. Well, thanks for sharing your background and your path to Brown with us. Before we get into the details of the technology you've been looking at, I wanted to ask you more of a philosophical question. So PFAS are regarded to be one of the most challenging environmental contaminants we deal with. What is your outlook on our ability to tackle this human-made contaminant with technology?
So I always think, I guess my philosophy is that, we're engineers, we have this challenge. It may take us some time, but we should try a lot of different things. Some things will fail or not work so well, and we continue that effort. So I think one way to look at it is now we're sort of in this what I would call a containment mode, where we're trying to contain the releases. They're widespread, but we're trying to figure out what the releases are, how fast they're moving, what sort of natural processes influence PFAS transport and fate. And then in terms of the technologies, I think the things I work on now are these in situ sequestration processes with basically, as you mentioned, ex situ materials, right? How do we get them into the ground? And so I'm drawing a lot on work I did previously with nanomaterials, and also with, I did a lot of work with surfactants or polymer flooding way back when we were trying to remove chlorinated solvents. So it builds upon I think we all are building upon our experiences and trying different things to be able to treat these compounds in situ. So I sort of think we're at this stage now where we're containment, ex situ treatment, we can do okay. But it's really expensive. And of course, the holy grail is to have an in situ treatment. I sort of think we might have to do some sort of combined remedies or treatment train where we might have a reaction that occurs to get some of the material And then we might have to still use a sequestration to contain that. But I think, in general, there's some resistance to sequestration because it doesn't destroy the compound. And so you still have to worry about, okay, well, we have this compound in the ground, now what do we do with it? But I still think, in a sense, as we progress and maybe we get technologies that can destroy the compounds, then we could combine those and use them together. And that'll probably be where we're headed, where it's not going to be one thing that treats these compounds. And the other thing to remember is it's a huge class of compounds. And right now we may only be looking at 6, or whatever is the main driver for at a site. But there may be compounds down the road that we learn are toxic that then we have to adjust and focus on those. So I think it will keep us busy for a while. And but I think there is potential to find remedies that can be applied in situ to break down and sequester these compounds. And that's sort of where we're headed. We're trying to do both. But as you know, it's really difficult to break these compounds down at room temperature.
Mm-hmm. Well, that's great perspective, and I think it really sets the stage for our conversation. I think I share your outlook on that. I think we can tackle PFAS. Technology will definitely play a role in that. Some things may work, others may not. Agree with you that it will take some time, but I would also foresee a lot of technologies, working together, complementing each other, perhaps in the treatment train. I'm a hydrogeologist, so I'm especially interested in in-situ remedies and what occurs, within the aquifer matrix. So I guess let's get into the technology itself that you've been working on in your team. So congratulations to you and your team on the recent publication in the Journal of Hazardous Materials. And that presentation is titled In-Situ Sequestration of Perfluoroalkyl Substances Using Polymer-Stabilized Ion Exchange Resin. So your research is novel to me in that it aims to deliver ion exchange resin to the subsurface as a permeable adsorptive material. Could you tell us about ion exchange resin? What is it, how it works?
So I am certainly not an ion exchange resin expert. And there, of course, are many people that have done that type of work for years and years. So we're basically, trying to sequester or attach the PFAS molecules onto the resin. And hopefully we hope that it's an irreversible process where they're basically adsorb or stick or exchange, and then they don't come back off. And of course, that's sort of where we're headed with it. So it would function as an exchange resin where you basically are focused on the polar group primarily within the PFAS. And so that's the idea, is really to take, in a sense, we're agnostic about the actual resin. Our goal is really show proof of concept that we can get these in the ground and have them attach onto the soil or aquifer material and sort of coat that and then form a permeable barrier. We don't want the particles to be trapped in the pore space, so we get no flow. That would, we have bypassing. So our goal is to get these particles to be small enough and stable enough that then they attach to the surface and sort of coat the surface of the grain or the soil, and in that way enhance the adsorptive capacity of the native material. So really, that's our focus. And there's obviously probably much better resins, or many different resins that could be used and it could be applied. And in a way, we picked one that was just being used in the literature and had been reported. There was some data out there, so we could run our own experiments and compare to that. But the key for us was to reduce the size and stabilize it in a polymer that then we could inject. And so, our key contribution is, how do we make a stable emulsion or suspension that then allows the material to be attached or adsorbed onto the soil grains and form this permeable barrier in situ. And that's really our contribution. And again, I'm not an ion exchange resin expert, so that's, there's probably there's people working on different resins, and the idea is how do you then take those and make them injectable?
Okay, that's actually where I wanted to head next. I'm really interested in, the process to prepare the resin, how it's delivered into the subsurface, etc. So you touched on it already, but could you please talk about how the ion exchange resin is prepared and delivered into the porous media?
All right, so we just start with off-the-shelf Amberlite 910. And so I have a little, you asked me to bring a little container. This is just the stuff we bought from Sigma Aldrich. If we open it up and look, you might be able to see inside. It's this little, that's what it is.
Mm-hmm.
Right. So its little beads are kind of stick together. This is problematic a little bit. So when we first started working with this, it's like, how do we get this stuff to, we want it in the 200 to 300 nanometer range. That's what the size we want. So what we do is, and it's described in the paper, and what we do is we basically ball grind it first, and then we put it in a blender, and then we sonicate it for a little while. And then we have a polymer, and again, we used off-the-shelf polymer. It's called Pluronic, and relatively non-toxic polymer. So the idea was we don't want to put something in the ground that's going to cause problems in terms of toxicity. There are probably other polymers one could use, but in our paper we just we don't try to keep the chemicals proprietary or anything. We explain the process and how we did it, and so we mix those together, and it forms a milky white solution, and it looks a little bit like cream, or maybe a little bit thinner than that. The idea is we don't want to have high viscosity, because we want some viscosity can help you in terms of delivery, but if you're in an unconfined aquifer and you have a high viscosity fluid, then you have to adjust your head. You're not going to get the stuff in unless you inject it at really high pressures, and we wanted it to be injectable at relatively low pressure and not at natural gradient, but flow rates that are reasonable, where we didn't have to pressurize the system to inject it and just disrupt everything. So that was our goal, to have a solution that was low viscosity, low particle size, so we could inject. And again, a lot of this work comes from work we've done with nanomaterials, such as iron oxides, and facilitating their transport with different polymers. So we've tried a lot of different polymers to enhance the mobility of nanoparticles in porous media. Things like xanthan gum, some non-ionic surfactants, anionic surfactants. We've tried a whole bunch of different surfactants. So part of the background to that was, how do we get particles into, injecting? We don't want them caking or blocking the pore space. And then how do we get them to move? And then we want them to attach. So, and also it's sort of a balance. You want them to attach, but not too much. So if they're really strongly attachment, like high deposition, then you clog the pores. You can have basically physical straining and too much attachment. And so it's a balance between we want attachment, but not too much. And so our idea is we want to create a radius of influence that might be a meter or two. And then you would inject and have these areas, and you could go along and create a barrier that's perpendicular flow.
Okay. Pardon?
Does that make sense?
Yeah, that makes absolute sense. And again, as a hydrogeologist, those are the things I'm particularly interested in. How do you deliver this type of material, into the aquifer matrix and into the pore space, without clogging it up, and in a way that doesn't necessarily have to be pressurized and can kind of maintain the natural conditions of the system. So that's why this topic was very exciting to me. And as I mentioned in the introduction, the theme of our podcast here is really focusing on either novel processes or especially novel materials. And although some of these materials are off the shelf, it's interesting what you've done to transform them in your especially with your background in nanomaterials and being able to deliver this material to the subsurface, which I agree in situ is very important for the remediation of PFAS. So we talked about what you need to do to the material, how you get it into the subsurface, and kind of the conditions you want to maintain. How were you able to assess, the delivery, the coating, the adhesion, of the ion exchange resin in the porous media after you got it in there?
Okay, Jeff, let me show you a few things from the paper that'll help present the size and how we analyzed the material and showed that, or demonstrated that it was attached onto the surface. So this is first thing, this is our DLS analysis, dynamic light scattering of the particles. And you'll see the main peak is at about 230 nanometers. We do have some larger particles here. You'll see a peak out here. When you do DLS, this can just be a few particles, but what it shows is that these are 3 different traces, and you can see we're pretty much in the 2 to 300 nanometer range, and that's really where we want to be. And the next figure I want to show you, this is in the supplementary information in the paper, but this shows you the distribution with distance from the inlet. So again, we were running column studies. This is sort of a proof of concept type of paper, but what's really nice about what this shows is we section the columns after the experiments, after we inject, and then we basically inject the solution, and then we go to the background or background synthetic groundwater or just a background electrolyte solution, and then flush out any residual. So this shows you the distribution of the carbon with distance, and you can see it's quite uniform across the length of the column. And this is what we want. We don't want a whole bunch in the front and then it drops down. So sometimes when you see particle distribution curves, you get a lot of attachment right near the inlet and then it drops way down. And this shows that we get a very uniform distribution of basically the beads within the porous medium. And last thing I want to show you that is probably the most convincing is we did some SEM imaging before and after. So this on the left is before. So this is a sand grain before treatment, and on the right is after treatment, and you can see all these little bumps on the surface. And so then, of course, the question is, are these really, the beads, or is that just you picked a different grain of soil and it is more bumpy than the other one? So, which is a good question, honestly. So what we did was then we run EDS. So this is a way to look at the elemental composition, and you can see these scans down below. And of course, we get, you get a lot of oxygen and different elements. But what I want to show you, this C is the carbon, and then you see it jumps way up on the right-hand side. So we basically scan this little zone for these, and you'll see that these are a lot of our elements here, but the carbon goes way up, and that demonstrates that what's on that surface are the beads, the carbon beads.
So yeah, I was interested in the microscopy, and I'm glad you shared that. So it's evident just by visual inspection that it goes from a much smoother surface to a much more textured surface, and then you get the additional proof of the carbon spiking up, if you will, when the material is adhered to the sand grain. So that's very interesting. I'm glad you could share that with us, Kurt. Thanks for sharing that information, Kurt. We've been talking about the process of delivering the ion exchange resin into the porous media for in-situ sequestration. We've talked about the importance of it, adhering to the matrix, and how you went about demonstrating that it's been delivered to the matrix and it's adhering there. Let's talk about the actual absorption and removal of PFAS by the resin. How was that tested in the laboratory?
Yeah, so we used 2 approaches, Jeff. The first is we did a traditional batch reactor test with the material adsorption isotherms. So that was the first test, and we used both individual PFAS and we used mixtures. So one thing we did that was a little bit different than what we've done in the past is, we use PFOA and PFAS, but then we did a mixture of 6. So we did those in batch. And then after that, we had a pretty good idea of the absorptive capacity. It was pretty high absorptive capacity. And that was, we used the material after we had prepared it, which means after we reduced the size. One thing that was interesting was that we increased capacity when we reduced size. So we didn't lose capacity when we, as we modified the material, which was something we were worried about. A lot of times when you reduce size, you do increase the absorptive capacity, but in some materials, that's not always the case, especially ones that are microporous. So that was the first step. And then what we did was we ran additional column studies where we delivered the material or delivered the resin first, and then we flushed with a little bit of background solution, and then we introduced the different individual or mixture of PFAS. So a lot of times what we'd start with is a more environmentally relevant concentration. So we'd initially inject about 10 micrograms per liter or 10 parts per billion, which is still on the high end, but it's more reasonable. And we'd inject that for a while, but then after about 15 or 20 pore volumes, when we weren't seeing anything break through or come out of the end of the column, we decided, well, this could take a long time based on the absorption isotherms. So we increased the concentrations quite a bit to the milligram per liter range. And so we were using, for the mixture, we were using 60 milligrams per liter total, 10 milligrams per liter of each of the 6. And then what we did was we proceeded to carry out the experiment until we saw breakthrough. And so we were trying to figure out what's the sort of the maximum capacity at these very high loadings to see if it matched what we observed in the batch absorption isotherms. And that was the approach there. Otherwise, we would have had to run for many, many, many pore volumes until we got breakthrough. And once we reached that max, or when the PFAS started to break through, we ran them until they reached about their influent concentration, and then we stopped the experiment. So that was how we tested the performance in the what we call dynamic or in the column systems, to see how the delivered material that was attached was working. And of course, we were worried about detachment, worried about competitive absorption, those types of things that can happen when you have these materials attached to it. So worry about the resin releasing. Does it release over time? We didn't observe that. And we also were interested to see about competitive absorption between the different PFAS substances or different PFAS species that we injected.
Well, that makes sense. So I guess transitioning here to my next question, and I think competitive absorption could play into this as well. What additional complexities could occur in the native aquifer materials, native groundwater that's impacted by industrial sources of PFAS? So if we're looking at applying this technology to an aquifer, what additional complexities could occur in the native aquifer materials, native groundwater, and that's impacted with industrial sources of PFAS?
Yeah, so there's a number of things, of course, we could investigate or look at that might be relevant at certain sites and not at others. But some of the key things are if you have some background material. So you could have natural organic matter occurring. Usually it's relatively low in groundwater, or you could have some type of other organic contaminant in your system. So you could have other organic species that are competing. And so we worry about that. We've started doing experiments to look at that, but that wasn't really covered in this paper because it was more of a proof of concept. We worry a little bit about ionic strength, as we always do. I think we worry about ionic strength. We ran at relatively, I would call medium ionic strength. We're at 10 millimolar, but you could have effects of different competing ionic species in your system. And the other thing is if you had different polymers in your system, you may or may not have polymers. They could also impact your performance. So we're looking at long-term stability of the resin. Does the resin stay attached, if you change solution conditions? So far, it seems to work pretty well. And the other thing is basically the competition between other organic species or other things in the background solution or in the native groundwater that, we're sort of might be competitive. The last thing we're sort of looking at, most of the materials we use so far are relatively clean, either reference, aquifer materials or aquifer materials that are primarily quartz sand. The ones that have the high permeability, we expect that's where most of the transport is, and that's where the least amount of absorptive capacity is. So the idea was you can get the biggest bang for your buck in those types of materials. But now we're transitioning to other types of aquifer materials, which are, finer materials. They might have other species in there that could either impact the injection, how well we can distribute the material, or impact the performance of the material once we get it injected. So that's sort of where we're headed. We want to know, long-term performance, how different system parameters could impact the performance of the resin. And I think these are all logical things that, anyone would say, well, what about this, Kurt? We need to look at that. And that's sort of where we're headed with it right now. The first paper was really like just to demonstrate that it has, pretty good potential to be a sequestration agent.
Do you have any research in progress or plan to answer or address those types of questions?
Yeah, we just got a new startup grant where we're going to look at the longer term performance of these injectable materials. So that's where we're headed with the new project, and that should be online pretty soon, hopefully by spring. But of course, we'll continue our work. We have a number of projects going on with this type of work, as most people do now, because PFAS is, pretty popular research topic these days. So we'll continue this work, but the idea is that we can look at these details and then try to run some, pilot scale field tests, partner with other people to run those types of tests using this type of technology.
Well, it's very exciting. Definitely always interested in how to take this type of research and bring it into the field and deploy it. So you've talked about that. This initial paper was a proof of concept, and the results look pretty promising and exciting. We understand that you're interested in looking at the long-term viability of the technology, and you have to look at some of those aquifer type considerations when you get out into the field. I'm also interested in scale. You just mentioned you'd be looking to pilot test it. How scalable would you see this technology in the future? What's your vision for its application to contaminated aquifers?
Yeah, so this is a great question, Jeff. So usually when I do work in the field, I like to partner with different groups. So there's basically two ways to look at this. One is we have to get the material small enough, and then adding it with a polymer is not too difficult to do in the field, or you could have a concentrated solution that you prepared and then diluted it once you got to the field. But the key thing, I think, is to how do we reduce the size? So how do we go from the beads that I showed you in the little container and get them down to 200 nanometers in a cost-effective way? So that, to me, is the major thing. And so I'm working with some of the suppliers now to sort of tackle that issue, if they're interested in developing in this direction. So you would have to set up a system to produce enough mass or enough of the material that you could then inject it. And so that's a critical step to moving this technology forward. I think the mixing with the polymer, once you get the size reduction and the injection process, will be not a huge challenge. I mean, we do that in the field now. Consultants do a lot of injections now. I think that part is pretty well established. So the key thing is, how do we get this material? How do we deliver it to the site, mix it, and then inject it? So I think that's where my focus is now in terms of making this more commercially viable, and then able to pilot test.
Yeah, my mind goes to a funnel and gate type system just to reduce the amount of volume that would need to be injected.
Right.
So, that might be one way to go as well.
Yeah. So my thoughts on, like, if you go to a funnel and gate, you can basically reduce the area that you need to treat or inject. You can almost do a trenching application, right? It's the size becomes less important because you could just inject. But sort of the holy grail of where I want to get to is that we can inject it in any place just through a regular injection well. Yeah, so we don't have to go to funnel and gate. We can inject it anywhere. That's sort of where I come from. Just my background is that's what I want. So, and that's, if you couldn't reduce the size, you get into a situation where you can't reduce the size and you just need to mix it into the soil, then probably a funnel and gate makes sense, because then you can trench out a little area. You can put it in, basically, you can inject it however you want to. You can do high pressure injection. You wouldn't necessarily need to have it flow with the natural groundwater. So I think to me, the funnel and gate's like a fallback if we can't get, the other part working. That you could still apply the technology, but you wouldn't have to worry so much about getting the size down and being able to inject it anywhere.
That's what I was thinking about, it along the same lines. Maybe as a fallback, it just allows you to kind of control the circumstances a little bit better. And I could almost see that, an extended gate where it's not a gate so much as it is a gauntlet of maybe complementary in-situ technologies. So just kind of thinking off the cuff here. But, I also see this also being complementary with just, basic soil scraping or soil removal. As we know, especially at AFFF type releases, a lot of mass is hung up in the shallow soil. It's held in the vadose due to interfacial absorption, but it continues to leach. So it's in some ways it's just depending on the size of that footprint, you can reduce that source strength, but then you still have the dissolved plume to deal with, and this sounds like it might be the perfect match. So it's very exciting, and I am sincerely interested in this subject. I'm really, a lot of my focus is on fate and transport, and the differential absorption of PFAS to the aquifer matrix, positively charged mineral surfaces and organic carbon. And to the extent we can sort of control that with this type of process and take, this, what's traditionally looked as an ex-situ treatment material, and put it in the subsurface, I think that's very exciting. That wraps up this episode of the Disrupting PFAS podcast. Thank you to Dr. Kurt Pennell of Brown University for joining us today. I'm your host, Jeff Hale, reminding you to never say forever.